Hey everyone, Mr. Murphy here. I'm going to continue with section 3.3, which is all about enzymes. It's another big one, as it's an important concept in understanding what's going on with biochemistry and biology. So, inside of our cells and inside of all living organisms, we have a whole bunch of proteins, as we discussed, and one class of protein is enzymes. Enzymes are catalysts, and what that means is that they speed up chemical reactions. They don't do anything that wouldn't otherwise occur. They're just facilitating the reaction to allow it to happen much more quickly and much more easily. So whenever we think about an enzyme that helps to build up a molecule or a different enzyme that helps to break something down, it's not that those molecules couldn't come together or break apart on their own. It's just that it probably would require very chance interactions or a very specific amount of heat energy and kinetic energy as these molecules smash together or break apart or whatever. And enzymes basically just grab onto molecules and situate them in just the right way in order to enable that to happen more quickly and more easily than waiting for it to happen by chance. So this is done by managing what we call the activation energy. Activation energy is just a simple way of thinking about how much energy is needed to make a chemical reaction occur. You have reactants, you have products, you need a certain amount of energy to get over this sort of hump in order to put that molecule into an unstable enough shape that it's willing to, for instance, break apart. So if I have a disaccharide like sucrose, that can break apart into glucose and fructose. That can happen by chance. If you get enough heat energy or whatever, they twist or pull in just the right way. You've got a nearby water molecule that's ready to give an OH and an H because of its own sort of push and pull. That can happen on its own. But it's not terribly likely to happen unless you have that energy available, like ready to go to smash these things together or whatever, in just the right way. And so enzymes are a way to manage this activation energy and to bring it down to a level that is more readily available within a biological organism. So in this example, even though the reactant, has this higher stored energy than the product, and therefore it may be chemically more stable for these molecules to break apart into their smaller pieces, it still needs some energy to do that just because, my example of a sucrose molecule, it's happy. All of the atoms are happy. They have full energy orbitals. They don't really want to break apart, even though they could break apart and be, happier from like a chemical standpoint. They're perfectly fine how they are. And so it needs energy to put it into that unstable sort of state where some chaotic things can happen and, you can change the form of the molecule. So this is just another representation of that. Enzymes do this by basically just putting the molecule or molecules in the right place in the right time. So here it says a molecule of sucrose in solution might hydrolyze in about 15 days. With sucrase present, that's a ase ending, means enzyme. Sucrase would be an enzyme that breaks down sucrose. That same reaction can occur in a single second. So massively speeding up the likelihood of a chemical reaction. And here's a representation of how it lowers that activation energy. Understanding this is pretty easy, but you got to think about it right. The enzyme has a very specific 3D shape, and that 3D shape has what's called an active site on it. There's nothing particularly special about an active site, except that active site has just the right arrangement of potential hydrogen bond regions that it matches a substrate that enzyme is there to work upon. And so by attracting the substrate into the enzyme molecule because of those hydrogen bonds, the cell is a chaotic place of molecules bouncing into each other. But those chemical reactions, or rather those chemical attractions between two molecules can mean that when things are smashing into each other, maybe they stay. And they stay because there's hydrogen bonds that sort of grab it and lock it in. So enzymes have a shape that matches chemically the shape of a substrate, and it brings it in and attracts it to the active site. So maybe we bring sucrose in and we attract it into our active site. Well, the active site is in just sort of the right arrangement that it allows for the separation of the sucrose monomers to occur more easily than they would just freely floating in the cytoplasm or wherever. And so and there's a little bit more to this. The enzyme can actually be sort of pulling physically on the molecule as well. We'll see that on a later slide, just to sort of put some strain and just to help the process to happen. Now, important to know is the enzyme is not in any way used up in this process, nor does the enzyme use any energy, like in terms of ATP or anything like that. The enzyme is purely based upon the fact that there are attractions and interactions between different molecules that can lead to sort of a docking of two molecules, and then also can lead to sort of a flexing or change in shape of these molecules based upon how they are being pushed or pulled by ionic bonds, van der Waals interactions, hydrophobic interactions, hydrogen bond interactions, any of the things we've talked about. So a way to think about this is the enzyme substrate complex, which is to say that when you have an enzyme here and you have a substrate here and they come together, we call this the enzyme substrate complex. That's when they are held together, and that's where the enzyme is not changing itself but is facilitating the reaction. This is a diagram which shows what I was alluding to before, the induced strain model of enzymes, where when that substrate comes in, the enzyme is a folded up protein. It has all those amino acids attracted to each other in all the ways. It's got that sort of dynamic but mostly stable shape. And now suddenly you bring this substrate in that has its own positives and negatives and interactions, and that's going to push and pull on those amino acids. So they may suddenly now start changing their shape to be attracted or repelled by that substrate. And an enzyme that utilizes this is one that because of that shape change, it actually puts some physical strain on that molecule and puts it into an unstable state, which then can allow for the reaction, that sucrose, to maybe break down into glucose and fructose a little more easily, because the glucose and fructose, while they're happy to be bonded to each other, now they're being sort of twisted or pulled or pushed in such a way that's kind of encouraging a breaking of that bond. So another way in which this can happen is that just simply by imagine a separate enzyme, maybe the opposite case, where it's taking a sucrose and a fructose and bringing them together to make sucrose. In this case, maybe it's not induced strain, but rather the enzyme has a particular shape. Let me go back to this diagram. If we take this in reverse, maybe this enzyme has a particular shape that lines up glucose here and fructose next to it in just the right way, where it's likely for them to bond together, to have condensation or dehydration synthesis occur. Ordinarily, a glucose and a fructose floating freely in the cell, they've got three-dimensional shapes. It's unlikely that they bump into each other at just the right orientation for the bond to occur. So an enzyme can actually line up the substrates just like that. Additionally, adding chemical groups here, the idea is that enzymes can allow the R groups to be directly involved in the reaction, that the different R groups here may be facilitating more complex changes within the molecule that situates it in such a way to encourage the reaction together or separation apart. That's a little more depth than what we're going to go into, so we don't need to worry about that, but just understand that, like everything in this class, that you can go layers deeper to understand more specific sort of sub-cases of how these things can function. So you can think of it like a baseball being caught by a catcher's mitt, and sort of how the shape changes when the ball hits and the catcher closes their hand around it. That's sort of like an enzyme catching its substrate and changing its shape. Just understand, the enzyme's not like, ooh, substrate, I'm going to change my shape. It's all chemistry. It's just simply the attraction of those things together can cause the shape change to occur. Here's another representation, reminding us that enzymes are made out of chains of amino acids. And here it can capture its substrates and align them or push them together or break them apart or whatever is happening here. Some important things just to remember, not from this slide, but in general. Enzymes typically only do one thing. So we have an enzyme that might build up larger polymers of carbohydrates, or we have an enzyme that breaks down polymers into monomers. They don't reverse freely. They don't do both things. And while some enzymes might have a little bit more specificity and need super specific glucose, not mannose, not fructose, There are other enzymes which are just a little more general in terms of the thing that they are attracting into their active site. We saw a slide on the last chapter which showed us mannose and galactose and glucose and fructose, and they all have really similar shapes. So you might have an enzyme that's attracted to a region that's actually the same on all those molecules. So in that way, some enzymes might be more specific or less specific than one another. But at the end of the day, an enzyme has a single job that it does. And that job may be I take any monomer of carbohydrates, or that job might be I take glucose only. Just depends on what its shape is actually attracting. So enzymes can need sort of help in this process, and that's where cofactors, which are inorganic ions, can sort of fit in with the active site to help to attract molecules, coenzymes, which can be other protein groups, which can add or remove chemical groups from the substrate. So it can be multiple enzymes sort of working together. That was sort of what I was talking about before as well. So these are just, again, understanding that biology is really complex. So while we like to have a really nice set of simple rules, understanding that in the background there's a lot of other sort of factors and things going on at any time, just try to be less surprised by that as the course goes on. And we'll learn to roll with it a little bit better as we think about these complex things. So there are a whole bunch of different types of cofactors and coenzymes. We don't need to memorize any of these, but if you see those terms come up, just understand that they are molecules which an organism may need to maintain within its cells in order to facilitate the function of enzymes. Some enzymes are very quick and rarely become saturated. Others are a little bit slower and may become saturated very quickly. The idea of saturation of enzymes is just the fact that enzymes are not instant. Chemistry happens fast, but chemistry does take time. And so when you have an abundance of substrates, but you don't have enough enzymes, you may max out on the rate of your reaction just because the enzymes are bumping into the substrates as fast as they can and catalyzing the reaction as fast as they can, but they are limited by the fact that they need to find the substrates and cause the reaction to occur. And that does take time. So you can have a situation where enzymes are maxed out on the rate of their activity based upon the fact that they're working as hard as they can. And so, we saw this with our enzyme activity, but as you continue to increase the concentration of substrate, you will max out at a certain point just because, again, your enzymes are working as hard as they can. Now, there's a lot more to maximum rate and some mathematical components to that. We don't need to worry about that. In the next section, we're going to be looking at the regulation of enzymes, which is to say the control of them and how that can allow for more nuanced sort of maintenance of homeostasis inside of the cell, both in a positive and a potentially negative way. If you have any questions, let me know.